Assalamu alaikum dear students, welcome to the course Principle in Animal Life 1. This is lecture number 47. The topic is glycolysis. Discussing agenda for the topic is glycolysis, and then how is glycolysis regulated. Lecture outcome. After watching and listening this lecture, students will know about one of the most ancient energy deriving pathways in living organisms, that is glycolysis. Glycolysis, it is also known as Embden-Meyerhof-Parnas pathway, and this pathway is regarded as the most ancient, or it is thought to have evolved very early in the evolution of life because of two reasons. First, this process occurs in almost every type of cell. It does not require oxygen, and the ancient environment was deficient in oxygen. Secondly, this process occurs in cytoplasm. It doesn't require mitochondria or any other organelle. So it can take place in prokaryotes as well as eukaryotes. Glycolysis harvests chemical energy by oxidizing glucose to pyruvate. So in this process, a 6-carbon molecule glucose is broken down into 2 molecules of pyruvic acid, each having 3 carbons. The word glycolysis means sugar splitting. Sugar refers to the glucose and splitting means the breakdown, and that is exactly what happens during this pathway. Glucose, a 6-carbon sugar, is split into 2 3-carbon sugars. These smaller sugars are then oxidized and their remaining atoms rearranged to form 2 molecules of pyruvate. In books, you will quite often see another terminology for this molecule, which is pyruvic acid. So, pyruvate is the ionized form of pyruvic acid. On the right side, you can see a short description of this whole process. It is a process where 10 chemical reactions are involved. Each reaction is catalyzed by enzymes. So these 10 reactions can be divided into 3 phases. The first phase is the energy investment phase. In that phase, the cell uses its ATPs in order to bring glucose to a form from which energy can be derived later. So this is the first phase of glycolysis, in which 2 ATP molecules are consumed. Then we have the second phase of glycolysis, where a single molecule is broken down into 2 molecules, and then this process proceeds further, and then comes the energy output or the energy payoff phase, where ATPs are produced. So you can see here, this is the first phase, which is the energy investment phase. Here 2 ATP molecules are used. The phosphate group provided by these ATP molecules is attached with the substrate, and the substrate molecules are rearranged so that energy can be derived from them later on in the process. And then 4 ATP molecules are produced through this process. Now 2 ATP molecules were used initially, and then 4 ATP molecules are produced. So the net ATP produced through this process is 2, and with that 2 pyruvate molecules and 2 water molecules are produced. Now along with these 2 high energy electron carriers, that is 2 NADH molecules, are also produced, and these NADH molecules will then deliver their electrons in the electron transport chain, and it will start the process of the electron transport chain. So as summarized in figure 1, glycolysis can be divided into two phases: the energy investment phase, where the cell uses its ATP, and the energy payoff phase, where the process produces ATP molecules. During the energy investment phase, the cell actually spends ATP. This investment is repaid with interest during the energy payoff phase, when ATP is produced by substrate level phosphorylation. So in this aerobic cellular respiration, we will study the production of ATPs via two mechanisms. The first is substrate level phosphorylation. Substrate level phosphorylation is a process in which one of the substrates, one of the intermediates of the pathway, provides a phosphate group to ADP and produces ATP. And then in electron transport chain, we will study another type of ATP production, which is oxidative phosphorylation, where the protons present in the inner membrane are pumped out into the intermembrane space, and as these protons return back into the inner lumen via the molecular motor or ATP synthase, so in this process ATP is produced. So the bulk of ATPs are produced through oxidative phosphorylation, whereas up to 4 ATPs are produced through substrate level phosphorylation in the breakdown of a single glucose molecule. The net energy yield for glycolysis per glucose molecules means a single glucose molecules enter into glycolysis, and the end product is 2 ATP molecules plus 2 NADH molecules plus 2 water molecules and 2 pyruvate molecules, each having 3 carbon. The 10 steps of the glycolytic pathway are shown in figure 2. All of the carbon originally present in glucose is accounted for in the 2 molecules of pyruvate. So glucose is a 6 carbon molecule, and at the end of glycolysis 2 pyruvate molecules are produced, which are having 3 carbon molecules each. So all the 6 carbon molecules are intact. No carbon is released as CO2 during glycolysis. Glycolysis occurs whether or not oxygen is present. So this process occurs both in the presence and in the absence of oxygen. Oxygen has no impact on this pathway. However, if oxygen is present, the chemical energy stored in pyruvate and NADH can be extracted by pyruvate oxidation, the citric acid cycle, and oxidative phosphorylation. Now once the glycolysis comes to its end and 2 pyruvate molecules are produced, so if oxygen is available, then these pyruvate molecules convert into acetyl coenzyme A through oxidative decarboxylation, and then this acetyl coenzyme A enters into Krebs cycle, and in Krebs cycle further NADH molecules are produced, and then in electron transport chain these NADH molecules deliver their electrons, and once again they are converted into NAD, and then they can return back to glycolysis and once more pick the electrons. But it all depends upon the presence of oxygen, because the last stage of electron transport chain requires oxygen. In case of unavailability of oxygen, the pyruvate molecules enters into other pathway which is referred to as fermentation, because the progression of glycolysis requires free NAD molecules. So the NAD molecules which were occupied in the breakdown of one glucose molecules needs to get rid of this electron so that they can return back into the NAD form, and the cycle continues. So if oxygen is available, then this pathway will enter into Krebs cycle and electron transport chain, and if oxygen is not available, then there will be fermentation. Two types of fermentation which we will study in the subsequent lectures. The first is lactic acid fermentation and the second is alcoholic fermentation. So right now we will only focus on glycolysis, actually what happens in this process, what are the different enzymes involved, and what are the different events which takes place in this reaction. So here you can see a graphical representation of the first 5 steps of glycolysis, where in the very first stage the glucose molecule, which is a 6-carbon molecule, glucose enters into the cell through GLUT transporter or glucose transporter. So once inside the cell, the glucose molecule is acted upon by hexokinase enzyme, and this is the phase where the first ATP is consumed. Hexokinase transfer a phosphate group from ATP to glucose, making it more chemically reactive. Remember, the first 4-5 reactions are the energy consuming process in which the ATP is consumed, and the purpose of these first 5 steps are to bring the glucose molecules or its intermediates into a form from which energy can be derived later. So the charged phosphate also traps the sugar in the cell. So you can see here one of the ATP molecule is consumed, and the phosphate group of the ATP molecule is given to the glucose molecule, and glucose becomes glucose 6-phosphate. In the next step, phosphoglycoisomerase, this enzyme converts glucose 6-phosphate into its isomer fructose 6-phosphate. So this is basically a process where the glucose is converted to its another isomer, and the molecules are rearranged. The groups around the ring are rearranged in this process. Now the third step, this is regarded as the most important step in glycolysis. Why? Because this is a rate limiting step, and the molecules formed in the first and second step, they can go back and form glucose again, and glucose can enter into other pathways. But once the cell completes the third step, then it is committed to go to the end and form pyruvate molecules. So this is an important rate limiting step, and here you will see a feed inhibition control. You have already studied the feedback inhibition in the previous lectures. Feedback inhibition is a process in which one of the products of the pathway acts as a control on the same pathway. For example, if ATP molecules are in abundance, then these ATP molecules, they bind to the allosteric site of this enzyme. As a result, when the ATP molecules binds to the allosteric site, then the active site configuration changes and it is no longer able to bind to the substrate. So this is basically a regulatory process, because ATP molecules will only bind to the allosteric site if they are in abundance, and if they are in abundance, then there is no need to break down further glucose. Then the glucose can be conserved in times where ATP is deficient. So when ATP is in abundance, then this ATP act as a control, it act as a regulator on that pathway. And remember, ATP is one of the products of this pathway. So we repeat this process again. Hexokinase uses ATP to phosphorylate glucose, increasing its energy potential. The second step is phosphoglucose isomerase. This enzyme converts glucose 6-phosphate to fructose 6-phosphate, and these two forms are referred to as isomers of one another. Now the third step once again uses ATP, and the opposite end of phosphofructophosphate, fructose 6-phosphate, increasing its potential energy. So you can see here it has 1 phosphate, and after the third step it has 2 phosphates, so the potential energy is further increased. Now the fourth step is catalyzed by the enzyme Aldolase, and this enzyme converts the fructose 1,6-bisphosphate into 2 molecules. So remember, in the beginning we said that there are 3 phases of glycolysis. The first is the energy investment phase, and this is the second phase. Now the 6-carbon molecule is split up into 2 molecules of 3 carbon each, and remember, these two are the isomers of each other, and this process is a reversible reaction. This dihydroxyacetone can convert into this product and vice versa, but energetically the favorable form is glyceraldehyde 3-phosphate. So from this step onwards we will mark 2 in each step, and 2 refers to 2 molecules of glyceraldehyde 3-phosphate. You can see here this is the cellular respiration. The 2 enzymes phosphofructokinase and hexokinase are present in the cytoplasm, whereas the other complexes are embedded in the inner membrane of the mitochondria. And you can see also here the molecular motor or ATP synthase, which synthesizes ATPs through oxidative phosphorylation. Now, just I said in the previous slides that from 5th reaction onwards there will be a figure of 2 against each molecule, because we are considering 2 molecules of glyceraldehyde 3-phosphate. So here in the 6th reaction, which is catalyzed by triose phosphate dehydrogenase enzyme, 2 NADH molecules are produced. And remember, this is the energy payoff phase where the energy is produced. In the 7th step 2 ATP molecules are produced. Once again, remember that we are dealing with 2 glyceraldehyde 3-phosphate molecules, so therefore this 2 is mentioned. 2 ATPs are produced in 7th step, and then further 2 ATPs are produced in the 10th step, and there is a liberation of water molecules, 2 water molecules in step 9. So this is the end product of glycolysis. What is the end product of glycolysis? 2 water molecules, 2 NADH molecules, and 4 ATPs. 4, but remember that in the energy investment phase, the cell used 2 of its ATPs, and now the second or the energy payoff phase has produced 4 ATPs. So we need to subtract those 2 ATPs which were consumed. So the net ATP production from glycolysis is 2 ATPs. 4 ATPs is the gross production, and if you subtract the 2 ATPs that were used in the first few steps, then the net production of ATP is 2 molecules of ATP, 2 molecules of water, 2 molecules of NADH, and most importantly 2 molecules of pyruvic acid or pyruvate. Each is 3 carbon. Now this is an important high energy electron carrier, and energy is also embedded in this molecule, and it was produced courtesy of glycolysis. Now when oxygen is available, this molecule will enter into electron transport chain and it will start this chain. Now what about the regulation of glycolysis? How glycolysis is regulated? So in this 2 or 3 slides we will study how glycolysis is regulated. An important advance in understanding how glycolysis is regulated occurred when biologists observed that high levels of ATP inhibit a key glycolytic enzyme called phosphofructokinase. Remember, this enzyme is used to catalyze the third step or the third reaction of glycolysis, and the basic purpose of glycolysis is to produce ATPs. So if ATPs are already in surplus amount, then there is no need for this pathway. Then the glucose molecules should be conserved, should be spared for a time when the energy reserves are deficient. So ATP molecules are already in abundance, then they will act via feedback inhibition and it will block this pathway. How they will block this pathway? They will attach to the allosteric site of phosphofructokinase. As a result, this enzyme will no longer be able to catalyze the third step. Phosphofructokinase catalyze reaction 3, the synthesis of fructose 1,6-bisphosphate from fructose 6-phosphate. This is a key step in the sequence. The product of step 1 and step 2 can be easily converted back to glucose by an array of enzymes before step 3. Then the sequence is not committed to glycolysis. So the product of step 1 and step 2, they can be converted back into glucose. But once the process crossed its third step, then it is committed to reach its end. So it is the major limiting reaction. But once fructose 1,6-bisphosphate is synthesized, it will not be converted back to glucose. Based on these observations, it makes sense that the pathway is regulated at the first committed step reaction, which is 3. Now how does cells do that? How does cells regulate this enzyme? ATP serves as a substrate for the addition of a phosphate to fructose 6-phosphate. So remember, in the energy investment phase, the first ATP was used in the reaction 1, and now the second ATP will be used in the reaction 3. In a vast majority of cases, increasing the concentration of a substrate would speed up the rate of a chemical reaction, but in this case it inhibits it. Why would ATP, a substrate that is required for the reaction, also serve as an inhibitor of the reaction? The answer lies in knowing that ATP is also the end product of the overall catabolic pathway. So recall that when an enzyme in a pathway is inhibited by the product of the reaction sequence, feedback inhibition occurs. So what is feedback inhibition? When one of the products of the pathway act as a regulator on this pathway. So here the basic purpose of glycolysis is to produce ATP. So ATP is one of the products of glycolysis, and then this product acts as a regulator on this pathway. When the product molecule is abundant, it can inhibit its own production by interfering with one of the reactions used to create the third reaction. Cells that are able to stop glycolytic reactions when ATP is abundant can conserve their stores of glucose for times when ATP is scarce. So ATP will only act as a regulator when it is in abundance, and the purpose of glycolysis is to produce ATP. So when ATP is already in abundance, then there is no need to further break down glucose, and therefore glucose should be conserved for the periods when there is energy shortage. As a result, feedback inhibition in glycolysis contributes to cellular homeostasis. How do high levels of the substrate inhibit the enzyme? Now, how the glucose act as a regulator, how ATP act as a regulator on the third step? As the figure shows, the phosphofructokinase has two distinct binding sites for ATP. So on the right side of your picture, you can see a phosphofructokinase which catalyzes the third step of glycolysis, and it has two binding sites for ATP, not one. The first binding site for ATP is the active site. When ATP is bound here, then this enzyme actively catalyzes the third step. Whereas if ATP is bound here, then this molecule, this enzyme is no longer able to catalyze the breakdown. So ATP can bind at the enzyme's active site where it is used to phosphorylate fructose 6-phosphate, or at a regulatory site where it turns off the enzyme's activity. So when it is bound here, when ATP is bound here, the enzyme's activity is hampered. It is no longer able to catalyze the reaction. Whereas if ATP binds here to the active site, then the enzyme can perform its function efficiently. So the key to feedback inhibition lies in the ability of the two sides to bind to ATP. When concentrations are low, ATP binds only to the active site, which has a greater affinity for ATP than does the regulatory site. So when ATP is in deficient amount, then there is a need to break down glucose and release more energy. So therefore ATP binds to this active site when it is in deficient amount, and this enzyme is active and keeps its function. Whereas if ATP is abundant, then there is no need to break down further glucose molecules. Then ATP arrives at the regulatory site, it binds here. As a result, the activity of this enzyme is blocked. So when ATP binds at this second location, the enzyme's conformation changes in a way that dramatically lowers the reaction rate at the active site. In phosphofructokinase, ATP serves as both a substrate and an allosteric regulator. To summarize, glycolysis starts with 1 6-carbon glucose molecules and ends with 2 3-carbon pyruvate molecules. The reaction occurs in cytoplasm, and the energy that is released is used to produce a net total of 2 ATP and 2 NADH. Remember, this is a net total, not a gross total. Gross total is 4, net total is 2. So now the question is, what happens to the pyruvate? So the cells derived 2 ATPs. 2 ATPs was a benefit of this cycle, but now remember that 2 NADH molecules were produced in this process and 2 pyruvate molecules were produced this process. So what will happen next? It will depend upon the presence or the absence of oxygen, and we will discuss that in subsequent lectures. Thank you very much. Allah Hafiz.